Hey guys, welcome back to Unstop Biology. So in today's video, we are going to start off with the structure of proteins. So we are going to basically discuss the structure of proteins in detail, as in how they first form the primary structure that converts into secondary and then into tertiary. And then we'll also study something about the different type of bonding in polymers. So basically, we studied in the last video. If you would have seen the last video, we studied that different amino acids in protein, they combine with the help of a peptide bond. So in the same way, different polymers have different bonding, right? So we'll study something about that as well. So also guys, if you have not gone the previous video of these chapters, we have discussed a lot already. So I would request you to go through all the previous videos if you have not. And if you have, yes, you will understand what I'm going to teach today. Right. So what I do is basically I do not repeat the previous topics again and again because that is nothing but simply wastage of time. What you can do is if you have missed any topic, you can just go back and check out my previous videos of this chapter, right? So basically, we'll start with structure of proteins. Now, as we discussed in the previous video, what are proteins? Proteins are polymer of amino acids. So different amino acids join together to form proteins, right? Now it can be a homopolymer that is of one single type of amino acids, and it can be a heteropolymer as well, which means basically different types of amino acids, right? So basically these are either homopolymer or heteropolymer containing strings of amino acids, right? So say the structure now, if we talk about the structure, what happens is that you start with a very simple structure when you start forming it, right? So say in case of proteins, it is structured at four levels. It is structured at four levels, wherein it starts with the primary structure, right? Now, if we say, so what we do basically is when we start study about structure, we start with a 2D structure, a two-dimensional structure, and then we get into more complex, which are 3D structures, right? So the sequence of amino acids, once this amino acid starts joining, that positional information in a protein, which is the first amino acid and then the second and then the third and so on, it goes on, right? So this is the safe, this is the first amino acid, this is the second, this is the third one, and then it keeps on going on till n, depending upon which kind of protein it is forming, right? So this structure, this 2D structure basically is nothing but the primary structure. This is what? This is the primary structure basically, right? So this structure that you see here, this, right? If we talk about this structure, basically what we're doing is here, we are taking this particular section. And if you check here, this is the structure, right? And this is nothing but, say, this is one amino acid. This is another amino acid. This is the third amino acid. And this is the fourth amino acid. So basically, if you consider this amino acid as 1, N, this is N minus 1, so it is growing towards the left hand side, N minus 1, N plus, N minus 2, and so on. And in the same way, it is growing towards the right hand side as N plus 1, N plus 2, and so on. Right. Now, what are these amino acids? This is serine, this is cysteine, this is tyrosine, an aromatic amino acid, and this is glutamic acid, negatively charged amino acid, right? So this is nothing but the primary structure. So basically, this structure that you see here is the primary structure of protein. So you imagine that the primary structure is nothing but a line, a simple line, right? It basically towards the left hand side and towards the right hand side, you will see all the amino acids aligned in a line. Now say there are two terminals. One is N terminal and the other one is C terminal. So here, as you can see, here this amino acid is ending at nitrogen and here it is ending at the carboxylic acid group. So basically O is here, C is here, right? So this is the C terminal and this one is the N terminal, right? So basically the last amino acid is the C terminal. So here the last one is here, right? And the first amino acid is the N terminal, right? So always remember there are two terminals. First one is the N terminal amino acid, last one is the C terminal amino acid, right? So basically now this protein thread does not exist throughout as an extended rigid rod. It starts folding, and first it folds into a helix somewhere, in the same manner as we studied in the case of DNA, right? Or if you can imagine a rounded staircase, a revolving staircase, right? So this thread, so the first structure is basically a thread. Now this thread starts coiling in the form of a helix, right? So this basically becomes a helical structure. And here what happens is that in protein, the helix, this rounding is right-handed, right? So this helix is right-handed helix, right? Now again, this helix basically is nothing but the secondary structure. This is the secondary structure, right? So now this helix, this right-handed helix in case of protein is the secondary structure. Now what happens after this? So now imagine that there is a thread first that coiled, and now this coiling starts forming, shape of a circular ball, right? So now basically it somewhat folds into a hollow woolen ball, right? And this folded ball structure is nothing but the tertiary structure. This is the tertiary structure. So now, after it forms this circular ball shape, this basically gives us a view of three dimension. This becomes a 3D structure, right? And why this, chain of amino acids is folding is because a lot of biological activities of proteins are carried out once they get folded into these tertiary structures. So basically, this structure is very, very necessary for all the biological activities of the proteins. Now, in some cases, in some proteins, what happens is that they even fold, you know, even in the form of a plate or a cube. So this structure now basically gets folded in the form of a cube or a plate, right? So this even more folded structure is known as quaternary structure. And I hope you would have heard of the protein hemoglobin, right? So hemoglobin, which our blood carries, has four subunits. It is four times folded. It is nothing but a quaternary structure, right? So basically, in the case of hemoglobin, what happens is they have two identical alpha and beta subunit of hemoglobin, and these are quaternary structures. So overall, if I have to simplify the structure of protein for you, what would I say? I would just say that there are four different structures depending upon the biological function of the protein. The first one is normal, a string, which is the primary structure. This gets coiled into a right-handed helix, which is the secondary structure. This forms a woolen ball, which is tertiary structure. And in some cases, now this even gets more coiled and forms a more complex structure, which is quaternary structure. It all depends on the biological activity of the protein. So this is all about the structure of proteins. Now, if you see here the diagram here as well, you'll realize that this is tertiary structure. Right. Now let's see what are different type of bondings which are formed in the case of different polymers. So in the case of proteins, the amino acids are linked with the peptide bond. Now peptide bond is basically formed when carboxyl group, right? So we saw that for the amino acid, one end is amino group, then we have something, and the other end is carboxyl group, right? So when this carboxyl group reacts with the amino group of the other amino acid, right? So here it reacts with the amino group and it eliminates the water and it forms a bond here. This is known as peptide bond. This bond is peptide bond, right? In the same way, in the case of polysaccharides, the monosaccharides are joined together by glycosidic glycosidic bond. Again, this bond is formed by dehydration. So this happens when two carbon atoms of adjacent, so this is the first monosaccharide, this is the second monosaccharide. It has all carbon atoms. So these carbon atoms, right, these two adjacent monosaccharides carbon atoms join together. Now this happens in the case of polysaccharides. This happens in the case of proteins. Now again in the case of nucleic acids, what happens in the case of nucleic acid? What happens that the phosphate group links the 3 carbon of one sugar of one nucleotide to the 5 carbon of, so there you have bond between the sugar molecules. So 3 carbon of one sugar is joined with 5 carbon of another sugar, right? So this is nothing but a phosphodiester bond, right? So it starts from phosphate and hydroxyl group of sugar, right? So basically in the case of nucleic acids as well, if you see the diagram of nucleic acid structure in the case of DNA, right? This is the secondary structure of DNA, right? Where it is coiled, right? So here, if you see, it is coiled, right? Right, this is one and this is the other strand, right? So if you see the structure, what they're showing is here this structure, right? If you see the structure here, you will see that this is one nucleotide, this is another nucleotide. Again, this is the third nucleotide and this is the fourth nucleotide. So here this bond that you see, this bond, right? This bond. So basically what is happening is that DNA exists as a double helix, right? Now the two strands are of polynucleotides and these run anti-parallel. So these basically run in the opposite direction, right? And the backbone of both these strands is sugar phosphate sugar chain. So you have one sugar, then phosphate, and then again sugar, right? And nitrogenous bases are more or less perpendicular to this backbone, right? So A and G, they basically complement, and in the same way C and T complement, right? So basically these are purines and pyrimidines, right? So they are joined together. Now in between C and G you have triple bond, and in between A and T you have double bond. Now at each step, what you'll see that the strand turns 336 degrees. So in one full turn, the helical strand would involve 10 steps or 10 base pairs. Now what happens is that each there would be the exact pitch, that particular one helix will be of 34. What? 34. Right. So basically the difference between each base pair would be 3.4. It is 3.4, right? So this form of DNA is basically bDNA, right? So basically in higher classes we study that there are almost a dozen form of DNAs which are named after the English alphabets, right? But this structure that you see here is bDNA, which is the most common form. Right. So, guys, this is all about the different types of structures of proteins and why is it necessary, and then the different types of bonds which are found in different types of biomacromolecules. So, I hope you understood this topic. In the next video, we are going to cover even more processes of pathways and more about enzymes. And guys, please do not forget to like it and share it with your friends. And if you have any doubt, please do not forget to message. You can either mention your doubts in comments, or you can also message me on WhatsApp. The number is given in the description of the video. Okay, guys, see you in the next video. Thank you. Bye bye.